So I'll wait for a couple of seconds and then we can start off with this webinar. Hello, everyone. Welcome to this virtual first session of the Discover Glow Tour. I'm really pleased to have you here and attending this first session. This is a combined webinar of Promega and Mimetas, the organ-on-a-chip company which is based in the Netherlands. And we have two speakers today. We have Terry Riss coming from Promega Corporation, and we have Flavio Bonanini from Mimetas. We will have two talks, so first starting with Terry Riss and then a talk of Flavio, and in between we will have a Q&A session. One thing I really want to add is, please, if you have any questions, please add them to the Q&A box, and we will try to respond to those questions during the Q&A session. And if you're not able to answer them during the Q&A session, we will respond back to you afterwards. So a thing I want to highlight is really add your name also during your question requests. So we are really able to respond back to you afterwards. The first speaker of today is Terry Riss. He is actually a global product manager from Promega Corporation, and he has already more than 30 years of experience in the management and development in cell health assays. And one last remark before I give the floor to Terry is that we will make the recording available after this webinar session is done. So we will certainly make that available for you. Thank you. And Terry, the floor is all yours.
Hear me okay?
Yeah.
Great. Okay, thank you everyone, and welcome to the webinar. Okay, my name is Terry Riss, as Lindsay said, and I'm a scientist at Promega, and I've been with the company for a long time, over 30 years now. And this is a timeline that I wanted to show. I don't expect you to be able to read all of the small print here, but it's just to highlight that Promega has a broad portfolio of cell-based assays. We started in 1990. I started the cell regulation lab at that time. And we have assays to measure live cells, dead cells, apoptotic cells, cells undergoing stress, reactive oxygen species. And more recently, we've developed a number of assays that are what are called real-time assays that enable you to record data in a kinetic mode. So we have assays that are based on absorbance, fluorescence, and luminescence as well. This also is a sort of a depiction of the portfolio of our assays. And the reason I put this up here is the assays that are shown with this little 3D and a square are those that have been validated to work with 3D model systems. And I'll go through some of those today, basically describe how some of those assays work, show some of the work that we did to validate those assays, and I'll mention some of the ones that Flavio will be talking about when he gives his presentation on the chip device. So in addition to having a broad spectrum of assays to choose from, scientists also have to choose which type of a 3D cell culture model they use. And there's a lot that goes into this decision. You can work with a a matrix-free environment, using individual spheroids, and there are examples of people using a 1536 well format where they can simply pipette cells into a ultra-low binding plate, enable the cells to form a spheroid in a matrix-free environment, add the test compound, adding the detection reagent, and everything is done in a homogeneous format in a single well. So that's the simple high throughput approach. On the other extreme end of the spectrum, there are examples of people using human on a chip or organ on a chip devices that may have as many as 10 different organoids connected in a microfluidic device that pumps the fluid through, and in some cases will even stretch the environment to enable the cells to experience stretch. So that's an example of something that's very complex and actually cost much more. And the disadvantage is you typically get, one chance. You have one sample to analyze with that high expense, high complexity example. So what you need to do, and in fact, so most researchers are somewhere in the middle. They are using either, more than one cell type or differentiating organoids to use, or using simple microfluidic devices like the one that Mehmetis will describe today. But when you choose, you have to, consider what is fit for purpose. In other words, the information you get, you have to ask yourself, is it worth the investment? You don't want to over-engineer your model system. And what do I mean by fit for purpose? Fit for purpose simply means designing an assay that's as simple as possible, but yet as complex as needed to be biologically relevant. Or another way to put it is, it's as complex as needed to get the answer to the experimental question that you're asking. Another thing to keep in mind is you need to consider both the assay method and the 3D model culture system to be part of what we call the model. So what's different about 3D cell culture? On the left, you see a field of fibroblasts, a monolayer of fibroblasts grown on plastic. And on the right, you see a spheroid that was grown in an ultra-low binding plate. The major difference as far as applying assays to these models is simply the size. That's a major consideration that you have to keep in mind. If you have a population of cells on a plastic surface, they may only be, 5 micrometers deep. So the distance that a reagent has to travel to be able to penetrate and lyse all of the cells is much different than in a spheroid. Even though you may have fewer cells per well using spheroids, using an individual spheroid, the distance the reagent has to travel, maybe, for a 500 micrometer spheroid, the reagent may have to travel 250 micrometers to be able to lyse all of the cells in that sample. The other thing that is of a consideration is the ability of light to penetrate. And that's especially true if you're using imaging to interrogate the spheroids. So there's an unmet need for assay methods that have been validated to interrogate markers in 3D culture. And most commercially available off-the-shelf assay reagents were originally designed using either cells in a monolayer or cells grown in suspension. And if you choose something that's off the shelf, you have to ask yourself, will the reagents effectively lyse all of the cells in that 3D structure? Or if you're using a small molecule probe like a fluorophore, will the reagent penetrate all the way to the center of the 3D structure? And even if the reagent or the probe does get to the center, will the signal be blocked or quenched before it reaches the detector? So those are all some things you need to consider when you're choosing an assay method. And those questions sort of prompted Promega to develop collaborations with a number of companies that are using these 3D models to provide, data that shows that we've evaluated some of our assays with these different chemistries. So as I go through the introduction part of the presentation today, I'll describe some of the approaches that we've used to overcome the challenges of interrogating these large 3D structures. I'll show the example of the ATP assay, which involved a reagent formulation. I'll mention a caste-based assay that in that case was just simply modifying the protocol. I'll also mention the approach of using a small molecule probe. And in this case, the CYP3A4, I think there's actually some data that Flavio will show using that assay approach. So the first method that we validated for use with 3D culture models was the ATP assay. So the cell titer glow cell viability assay is the name of the original product that we had to measure ATP as a marker of viability. And the reason we chose that system is because it's the most widely used high throughput screening assay for viable cells. It's the fastest and by far the most sensitive method for detecting viability with a plate reader. The reagent has a strong lytic capacity, and what I mean by that is it has strong detergents to lyse the structures, to lyse the cells, and that the product also has a stabilized luciferase that we know was not affected by strong detergents. And the assay also can be multiplexed with DNA staining to measure dead cells. So I'll show you that multiplexing approach. In other words, using an orthogonal method to measure an endpoint that you can use sort of as an internal control. So I will describe to you how the ATP assay and the DNA staining assays work to measure live cells, dead cells, and the value of using these in a multiplex format. So first with the ATP assay, the general concept here is that viable cells contain ATP, and as soon as cells die, the ATP disappears. You know, the endogenous ATPases chew up the ATP and the cells lose the ability to synthesize more ATP. When you do an ATP assay, you add, in this case, the cell titer glow reagent, and the reagent contains a strong detergent in a lysis solution. And when you add that reagent, it immediately lyses the cells to release the ATP. The reagent also has ATPase inhibitors to stabilize the ATP and luciferin and luciferase to drive the luminescent reaction. So very simply, live cells give you a luminescent signal. The DNA staining, the DNA dye method, works a little bit differently. There's a small molecule, it's actually an asymmetric cyanine dye. It's called CellToxScreen. It's added directly to cells in culture, and that dye is not permeable to live cells. So it doesn't stain the live cells, but when cells die and lose membrane integrity, the dye can enter the cells and stain the nucleus screen. So it binds to ATP. And what I'll show you now is a critical experiment that Promega did that actually prompted much of our effort to begin validating these assays. And this has to do with observing the lytic efficiency of the ATP detection reagents. And the way that we did this experiment is we grew spheroids to about 350 micrometers in diameter, and then we added a combination of reagents. We added the DNA dye directly to the ATP detection reagent so we could add them simultaneously to the organoids that we were studying, or the spheroids, I should say, that we were studying. And then we followed the original protocol with the original cell titer glow assay, which was to simply incubate for 10 minutes, and then we took pictures using confocal microscopy. And you can see here the results. Actually, the image on the right was a spheroid that was treated with the cell titer glow reagent in combination with the DNA binding dye. And what you're looking at here is if the cells have been lysed, they will be stained with the fluorescent DNA binding dye. Okay, so everything was apparently lysed that was treated with the cell titer glow reagent. And this was actually compared to another competitor's reagent, and the one on the left simply shows an example of a reagent that does not effectively lyse all of the cells in that 3D structure. So that sort of raised a red flag for us, and suggested that we needed to do some validation of assays to make sure that we were lysing all of the cells. We actually carried the experiment out further, looking at spheroids of different sizes, extending out to 600 micrometers in diameter. You can see the image here, and we actually found that there were limits to the ability of the original cell titer glow reagent to lyse all of the cells in these spheres. And what we did was an experiment that compared acid extraction of ATP to the detergent extraction method that's present in the cell titer glow reagent. So it turns out that acid extraction is sort of the gold standard for extracting nucleotides from tissue. And the way that it works is you simply homogenize the tissue with a strong acid like trichloroacetic acid, and then you can neutralize that solution with a buffer, and then use that to record luminescence with a standard luminescent assay. And you can see on the graph on the right side here that when spheroids started to get above about 400 micrometers in diameter, the efficiency of recovery of ATP started to drop using the detergent extraction method. And you can actually see in the image on the left here that it suggested that not all of the cells were being lysed with the original version of the cell titer glow reagent. Now, I don't have time to go into all of the details, but those data led Promega to work to reformulate that ATP detection reagent into something that we now call the cell titer glow 3D cell viability assay. And to summarize some of the work, the reagent basically has a higher concentration of detergents to more effectively lyse all of the cells on these large 3D structures. And in addition, we modified the protocol to incorporate more physical disruption by using a plate shaker that would shake the sample for 5 minutes, and then also a longer incubation time in the presence of that lysis solution, up to a half an hour before you recorded the luminescent data. So after we did that, we demonstrated that this new version of the cell titer glow 3D cell viability assay, works well with a number of different 3D model systems, including cells embedded in hydrogels. This is an example of using orthogonal methods to measure endpoints. And these orthogonal assays can be used as a control. And in this case, what's shown is spheroids that were treated with different concentrations of panobinostat, just as a model toxin. And the DNA dye fluorescence was recorded first. And you can see the green line with higher concentrations of panobinostat. You see more and more green fluorescence, indicating the accumulation of dead cells. And then after the fluorescence was recorded, you add the ATP detection reagent, and you can see a decrease in the luminescence signal, indicating fewer viable cells. So the increase in the number of dead cells corresponds to a decrease in the number of live cells. So these orthogonal approaches can be used to help you confirm that your assays are working correctly. So what happens if your sample or the thing you're trying to measure is not stable to detergents? And the example I'll show you or mention here is the Caspase Glow 3.7 assay. We knew from our original efforts to develop the Caspase Glow assay that the caspase enzyme itself is not stable in very high concentrations of detergent. So when we attempted to apply this product directly to spheroids grown in culture, we could see that there were some issues with lysing the cells. So the challenge here, of course, is that we couldn't just dump in a lot more detergent to the Caspase Glow reagent because it would destroy the caspase enzyme. Even though the luciferase itself was a stable version that was not affected, we knew that we couldn't just add a lot more detergent. So again, in the interest of time and to make a long story short here, the result was that we simply modified the protocol of the Caspase Glow reagent or the Caspase Glow product to form what was called the Caspase Glow 3.7 3D assay. And the protocol was modified, again, to incorporate more physical disruption. So we shake the plates at 500 RPMs for 30 seconds prior to letting them just incubate for a longer period of time. In this case, it's recommended at least 30 minutes before you record the luminescent signal. So again, an example of not doing a reformulation, but modifying the protocol to address lysing 3D structures. So some of the general recommendations we have, if you're using an off-the-shelf assay, is if possible, simply change the detergent formulation, and that will help you lyse all of the cells. Or you can simply increase the physical disruption, as I showed you, or simply incubate the samples for a longer period of time in the presence of lysis buffer. So this is just a general set of recommendations you can follow. So another approach is to use a small molecule probe. So something that's able to penetrate into the middle of a spheroid or a large 3D structure, and it can overcome the limitations of attempting to lyse all of those cells. And some examples of that, I already showed you the DNA binding dye. It's able to penetrate spheroids. Other approaches that I'll mention are the P450 glow substrate. This is a modified luciferin molecule. It's called luciferin IPA. It's able to penetrate cells and get reduced, or get modified, I should say, into generating luciferin, which can be used. And I think Flavio is actually going to show some data using the CYP3A4 assay. And another approach to measure viability in this case is the real-time glow assay, and I'll show you how that one works. But first, going back to the P450 assay, the way that assay works is a substrate, which is a modified luciferin, is simply added to the cell culture, and that proluciferin can penetrate cells and get changed chemically by the CYP3A4 enzyme to generate luciferin. And then to run the assay, you simply sample an aliquot of the culture medium and move it to an assay plate, and then add a luciferin detection reagent and record luminescence. So this is an example of relying on the ability of a small molecule to penetrate cells, and then the luciferin again is permeable, so you can simply sample an aliquot of the culture medium to run the assay. The real-time glow MT cell viability assay works in a similar fashion. In this case, the luciferase that is used in this case is an engineered luciferase that came from shrimp. So in this case, the substrate is different. It's not luciferin, but instead it's a modified cilantrazine. So the product, or the reagent, the reagent in this case includes the pro substrate and the nanoluc luciferase. When you run the reaction, you simply add the real-time glow reagent directly to cells in culture. The pro substrate is taken up by live cells, and it's converted, or actually it's a reduction reaction, to generate a substrate that can be used by the nanoluc luciferase. As long as you have a viable population of cells in the well, you will be generating a luminescent signal. You can add this reagent once at time 0, and depending on the cell number, you can record kinetic data for as long as 3 days. So this slide actually shows the results of a 3-day experiment where cells, in this case, were treated with different concentrations of thapsigargan as a model toxin, and the plate was actually put into a plate reader that has an environmental chamber, and it was programmed to record once every hour. So you can see the vehicle control, or the lowest concentrations of toxin. Over that 3-day period, you have an increase in the luminescent signal. And the wells that had the highest concentration of toxin, you see a decrease in the luminescence, suggesting that those cells are dying over this 3-day period. So you can record this kinetic data in real time from the same sample by adding that reagent once at time 0. And of course, we've demonstrated that this real-time glow MT cell viability assay also works to estimate the viability of cells in spheroids. And we grew spheroids to different diameters here and applied this real-time glow assay to demonstrate that that small molecule probe approach also works. And by the way, if you were to do an ATP assay, you would see the same trend line with increasing diameters of the spheroids. So I also want to mention metabolite assays because Flavio is actually going to show some data from their chip device and measuring metabolites, and different metabolites that can be converted by dehydrogenase enzymes to generate either NADH or NADPH. Those metabolites can be detected using a luminescent assay chemistry. And this is just an example, just to show how the assays work. So if you want to detect a metabolite in cell culture medium, if you can match it with a dehydrogenase enzyme that's specific for that metabolite, you can create a reagent that contains the dehydrogenase enzyme and NAD, and then when you add the metabolite or you sample a culture medium, what you do is you convert that metabolite to some product, and in the process, you generate NADH or NADPH, depending on which enzyme you use. And then the reducing equivalents from that NADH can be used to drive a reaction to convert a proluciferin into luciferin. So in this case, the proluciferin is not a substrate for the luciferase until this reduction or this reductase enzymatic reaction occurs to generate luciferin, which then we can use to drive a luminescent assay. And there's a wide variety of metabolite assays that we have available, and I think Flavio today will show some data from measuring glucose and glycogen. So the summary from my introduction before Flavio speaks is that keep in mind that there's a wide variety of cell health assays available, and there's also a wide variety of 3D culture methods available, and you have to make some compromises when you choose which assay to use. In other words, when you choose a fit-for-purpose assay, you don't want to use something that's overly complex, that would be too expensive, and has a low throughput. Also keep in mind that most of the commercially available in vitro cell-based assays were originally designed using monolayers of cells, and you should validate in your own lab whether or not those assays work with your particular 3D model system. Some of the major considerations you have to think about is the size or the mass of the 3D structure, because that can limit efficient lysis of all the cells if you're using a lytic assay, and it can also affect the ability of light to penetrate or be detected if you're using an imaging device. When you're applying assays to 3D models, some of the recommended first steps we recommend if you have to modify the assay is to optimize the lysis buffer formulation, or simply increase the physical disruption by shaking the sample, or increasing the incubation time with the lysis solution before you record the signal. And if those don't work, you also might consider changing the assay format to use a diffusible small molecule probe, like I showed you with the CYP3A4 assay or the real-time glow viability assay. Also, keep in mind the availability of multiplexing orthogonal methods to help you validate the assay performance. Sometimes it's difficult to come up with an appropriate control to determine whether or not the assay chemistry itself is working, but you can use those orthogonal approaches. And the example I showed you was to correlate a decrease in viable cells with an increase in the number of dead cells. And of course, the examples that I showed you today, most of those were using spheroids of, in many cases, they were HCT116 cells. And because the nature of these 3D models change over time in culture, or different cell types have different physical properties when they form these 3D structures, it's highly recommended that you validate the assay with your individual model system. I also included this slide that has links covering several topics or more detail about the topics that I didn't have time to go into today. So you should be able to go to these links and find additional information. And with that, I will entertain some questions. So Lindsay, if you see questions coming in.
Yeah, thank you, Terry. We had a question about the real-time glow assay. If we have any cytotoxicity data on the kinetic experiment.
When you say cytotoxicity data, the 3-day experiment that was shown demonstrated a decrease in the luminescence signal with higher concentrations of that toxin. So that decrease in the luminescence signal is an example of cytotoxicity measured with that kinetic approach.
Okay, thank you, Terry. I hope that answers the question of the person. If not, please put in another question if you want.
Yeah, if they just want to clarify the question, if they send, you know, send in the question, we can provide a more detailed answer.
Yeah, indeed. So the person is certainly, if you have extra information needed, please reach out. Then I think we can move to Flavio. So Flavio Bonanini from Mimetas will give the second part of the session today, and he is actually working as a researcher at Mimetas, and he's working on liver models and actually focusing on inherited metabolic disorders. So I'm really happy to have Flavio here in the room, and I give the floor to you, Flavio.
Thank you. Can you see my slide and hear me well?
We still see Terry, but we will move to your, yeah.
I see Flavio's slides.
Yeah.
Okay,
indeed.
Awesome. Well, first of all, thank you very much for attending today and for the invitation to present at this joint webinar. I'm trying to switch the slide. Okay, let's do it like this. So,
thank you.
Yeah. I studied at ETH biomedical engineering, and now I'm an early stage researcher at Mimetas, which is a biotech company located in Leiden, working under the consortium Polymer. So today I'd like to give an introduction to the Organoplate platform, some opportunities and the challenges of working with organ on chips, and some examples of application of Promega assays in the organoplate models, focusing especially on tubular models, but also looking at spheroid and organoid models. So we've seen from Terry that often increasing the complexity of your cell culture might come at a cost of its throughput. So Mimetas' endeavor is to mitigate this by producing high throughput organ-on-chip culturing devices. And these are called the organoplates. And essentially they are microtiter plates that instead of having normal wells underneath, they actually host individual chip units, which are microfluidic, singular microfluidic devices. These systems are membrane-free. They can be perfused with fluid without the use of any pump. And they allow the establishment of 3D cell cultures, also in complex arrangements with other cell types. And because the bottom of the plate is actually a glass plate, these cultures are, or these devices are excellent for imaging purposes. So how do organoplates work? So everything revolves around the invention and application of the phase guide. So a phase guide is essentially a little ridge that can be introduced in between two slides. And what this ridge does, it prevents a fluid from one side to overflow and permeate the other side of this ridge. So by doing so, it's possible to pattern very carefully microfluidic channels with different type of fluids. And often what we use is, for example, extracellular matrix. So this allows us to create interfaces between cells and extracellular matrix without the use of any membranes or artificial barriers. So depending on the organoplate design, it's possible to culture cells in a variety of different arrangements. So as I mentioned before, the most used one is a combination between a gel compartment and a perfusable compartments, like just with medium culture. So what's possible is to introduce cells in different arrangements, for example, by embed them in the gel already. Or it's possible to line cells on this extracellular matrix, which then form tubular structures. Depending on the design, it's possible to even expand this. So we have, for example, here the 2-lane or the 3-lane. And the 3-lane is essentially the 2-lane just with one additional channel, again defined by the introduction of a phase guide, which expands the variety of different architecture that it's possible to build. So with this technology, Mimetas developed a variety of different organ on chips and biological models. Many of them are barrier tissues. So the organoplate allows us to produce these very nice tubular structures, and I will look at an example later. Organoplates has always also been used to culture complex co-cultures, for example, blood-brain barriers, or to integrate the immune component, to look at migration of immune cells. And recently, Mimetas developed a new design which allows the combination of tubular structures and spheroids or organoids, which can be applied, for example, in the field of tissue vascularization, and is called the organoplate graft, and we'll see an example later. So an organoplate or an organ on chip gives a lot of opportunities. So these are complex culturing devices, which aim at integrating different physiological parameters that are often not considered in standard 2D cell culturing, which are, for example, the flow, the composition of an extracellular matrix, the architecture and polarization of the cells or of the tissue, the cellular composition, and the 3-dimensional environment. When we do organ on chips, we often miniaturize these systems. So this allows us to parallelize and increase the throughput and have many replicates of the same organ chip, with obvious advantages also in terms of reagent consumptions. So because of the glass bottom, organoplates are excellent for imaging, and since they're based on a 384 well plate format, they're actually they can be automated in their handling. However, miniaturizing cell culture also comes with some challenges that we've seen already. So 3D structures, Terry mentioned the accessibility of cells to reagents, and this is especially relevant in lytic assays. We generally have low number of cells, so we actually need sensitive assays that allow us to detect metabolites or biological events. Also, in this organ on chips, different cell types might be located at different places in the chip. So all of these considerations need to be taken into account when interrogating these cultures. And Promega offers a wide range of kits, and some of them we use routinely at Mimetas, and they're excellent because of their properties. So I'd like to start with one of these tubular structures. So what you see here is confocal imaging of one of our gut models. So this is composed of cells that are of primary source, grown as gut organoids, and then seated in the organoplate. So we can look at it quite in some detail. Here, for example, seeing all the cellular junctions between the enterocytes, and 3D reconstruction shows us this nice tubular formation. An important aspect of these tubular models is that they polarize the way they polarize in vivo as well. So the gut has an apical and a basolateral side. And by designing the culture as such, it's possible to polarize the cells in the organoplate in a way that resembles the in vivo situation. Furthermore, it's possible to access the apical side of these tubular structures because these are connected between inlets and outlets of this microfluidic channels. So here, for example, we see again this gut organoid model, and with the expression of esrin, which is expressed in microvilli on the apical side of the structure, and also the presence of different cell types that are also present in the in vivo gut, for example, goblet cells or Paneth cells. So all of this we can measure nicely and see nicely with optical imaging. However, Mimetas also develops understandable assays on how to interrogate these cultures. And when it comes to the gut, one very important aspect is the integrity of the barrier. So there are compounds that might compromise the integrity of the gut barrier that could eventually have some clinical scenarios. The way we do this is again by optical analysis, and essentially we add a dye on the apical side of this tube, and we take pictures at the regular intervals and look how fast this dye is able to permeate through the barrier. And in this case, we see that by adding a compound that induces leakage, we see the leakage of the dye very nicely correlating with the time, but especially with the amount of compound that has been administered. This can all be quantified, giving a leakage score. Mimetas also went one step ahead, and we can now measure the barrier integrity using TEER. So this is transepithelial electrical resistance, and essentially is a method to measure the permeability of a structure electrically. So high TEER would correspond to a good barrier, TEER to a leaky barrier. And organoTEER is essentially a lid with the electrodes that will insert themselves into the different wells of the organoplates, which allows us to measure organoTEER from each individual chip unit over time as well. For example, here we see a TEER of a gut model going up with the day of culture after seeding, indicating that the development of the barrier over time. So we can do all of this, but of course, some aspects of gut biology are still to be addressed. And one thing I would like to mention today is CYP3A4. So CYP3A4 is a drug metabolizing enzyme, especially expressed in the liver. However, it's of high relevance also in the gut. And CYP3A4 is responsible to detoxify things. And one pharmaceutical aspect that is often asked is whether a compound can induce CYP3A4 expression. And we can assess this by using the Promega CYP3A4 with luciferin IPA kit. So as Terry mentioned, luciferin IPA is this modified luciferin, which by CYP3A4 is converted into luciferin, which in turn then generates light. So the kit provides all the reagents that you need, and essentially this follows an incubation time and a luminescent measurement. Using this assay, we can, for example, look at the induction of CYP3A4 in these gut models. Here, for example, a Keiko tube or an organoid. And what we see nicely is an induction of CYP3A4 by adding vitamin D, especially in the organoid model. Whereas this activity is shut down with the addition of chemical inhibitors. So there are some technical considerations, as we've heard before, when it comes to performing these assays, which is the optimal incubation time, the accessibility of the substrate to the cells, and it's often also recommended to normalize to the cell count or to a cell viability readout, for example, the cell titer glow. I would like to jump now to a different type of culture, introducing the organoplate graft. So the organoplate graft is essentially a 3-lane, a modified 3-lane, which allows the formation of endothelium tubes on the sides. And by induction of angiogenic sprouting, we can then form vascular beds, which in turn can be used to engraft tissue and organoids. And here a video of this process taking place in which these vessels connect and vascularize this 3D liver spheroid. So again, using imaging, we can assess the morphology of the cultures, but also the perfusability. So here, similar as with the barrier integrity assay, we track the diffusion of this dye, and here in this case from left to right through this vascular structure. So we can assess a nice morphology and investigate everything at the cellular level. However, again, sometimes biological questions really needs a sensitive biochemical assay. One of the simplest is viability. So we've heard before the use of cell titer glow, which essentially lyses the cell structure to release ATP, which in turn is measured by using light. This is a one-step reaction, and the kit provides everything that you need. However, we've heard how important it is to optimize the protocol. So we apply this assay to the organoplate graft. And one of the first things that we did when we were validating this assay is to look at the different incubation times. So here we see spheroids that have been placed in the organoplate graft over different time points. And we can see that for a smaller spheroid, the signal seems to be stable already after 5-10 minutes. However, in the case of using 20,000 cells per spheroid, we still see a steady increase of the signal even after 25 minutes. So this really depends on your cell type and the culture type. But eventually, by optimizing it, then it's possible to perform very nice sensitive readouts. So because we have these different cell types in different compartments, we also investigate the potential of this assay to actually give us relevant biological information on the different compartments of the chip. So what you see here is a dose response of a toxin, and in green, the dose response in the graft chamber, and in blue, the dose response in the perfusion lane, indicating that there is indeed a different biological response to this compound, depending on the different cell type that is used. Cell titer glow has been validated internally, giving a Z prime factor of 0.86, and for those of you that are familiar, this indicates an excellent assay also for screening applications. So one assay that I am particularly interested is energy homeostasis in the liver. So one biological aspect that I want to capture using liver cells is gluconeogenesis. So gluconeogenesis is essentially the release of a newly synthesized glucose by liver cells in an attempt to supply the blood with glucose when during periods of starvation. And the challenge that I had was that I use very little amount of cells, so it's very hard to measure actually glucose because it's such in a low concentration because the baseline is 0. So Promega sells glucose glow, which is actually excellent in detecting this because it's very sensitive, and it relies on this NADH and NAD plus redox, which again generates light. The kit again is very easy to use. There's all the reagents are contained, and essentially it's sampling out from the plate and measuring, for example, at different time points. So this is what I did, for example, in this experiment in which I starved these liver spheroids for 24 hours, and then I measured the relative glucose output in the medium, showing the nice sensitivity of this assay to capture this biological event in miniaturized culture. Another aspect that I'm very interested in is glycogen. So glycogen is essentially glucose molecules bound together as a storage unit. So to measure glycogen, we can measure glucose. And glucose, again, similar as before, can be measured with this process, which gives a luminescent signal. However, to measure glycogen, we need to add one further step, which is the lysis of the cells, because glycogen is stored inside of hepatocytes. And furthermore, we also need to convert glycogen into glucose. And I've been lucky to receive from Promega an experimental kit which is called Glycogen Glow. And what this kit provides is all of the reagents besides, for example, the lysis buffer. So what I can see with this kit is that in this case, we have two conditions in which these liver cells have been either fed or starved. And if I add glycoamylase, which is this first enzyme that converts glycogen to glucose, I see a very high signal for the fed condition, whereas starved, I don't see anything anymore. So this black bar represents the glycogen amount, because if I don't add glycoamylase, which is indicated by the green bar, the signal is much lower. So these are some examples on how mimetas can use the organoplate, but also how it can amplify its biological relevance by addressing different biological aspects of its cultures. Using Promega assays always, especially when it comes to organ-on-chip or alternative cell culturing devices, there's usually always a little bit of optimization, for example, in incubation time or the sampling strategy. And the luminescent assay are actually excellent in providing us the necessary sensitivity to measure biological aspects from these organ-on-chips. The organoplate itself also comes with its own features and benefits. Some of them I mentioned before, with the different accessibility, for example, apical and basal access. There's no PDMS, excellent imaging quality, low compound absorption, and so forth. I come to the end of my presentation. I'd like to thank the people that contributed to this work, especially Dorota, Kinga, Sarah, Sander, Maria, Marin, and Philip, and also all the Mimetas team, and especially my consortium. And I think this is the last slide. I'd like to thank you very much for the attention, and feel free to ask any questions. Thank you.
Thank you, Flavio. I'm just looking if there are any questions coming in, so please feel free to ask any question to Flavio. In the meantime, I also had some extra questions for Terry, so maybe we can also come back to those questions first, and then we can come back to some questions for you, Flavio. Terry, I had a question about how far can luciferin penetrate in 3D spheroids, and how does luminescent signal is in spheroids if luciferase is already expressed by the cells and is not part of the reagents and lysis is not performed. Would the luciferin IPA approach would work as well, depending on the size of the spheroids? You may. Oh, I have to unmute you. Sorry. Wait there. That's strange.
Can you hear me now?
Yeah, we can hear you now. Yeah.
Okay.
Did you hear the question properly?
Yes, I did. So first of all, to answer the question about how far can luciferin or a substrate, a small molecule substrate, penetrate into a 3D structure, that would depend probably entirely on the cell type that was used, or whether or not there was matrix that was used to form the 3D structure. And it would be very difficult actually to determine how far it can go in, because it's not like, you know, your ability to detect it. It's not something that you can detect with like immunohistochemistry. You'd have to rely on some biochemical method to do that. And after the molecule penetrates in, you know, there's not really a good way to detect it. So what we do know is that we can see appropriate responses when we apply these small molecule probes to those 3D model systems. So it simply suggests that, you know, the probe can penetrate in and then be converted to the luciferin, which then, you know, diffuses back out into the medium and can be detected. So I hope that answered the question. And basically, I don't know the answer because it's very difficult to determine, you know, exact location of when these, you know, small molecules diffuse into these large structures.
And maybe also the second part of the question, Terry. So how does the luminescent signal is interpreted if luciferase is already expressed by the cells and is not part of the reagents and lysis is not performed?
Another good question. You can have cells that express luciferase, either the firefly luciferase or the shrimp luciferase, and you can have those cells expressing that enzyme, and you can apply the substrates to the cultures, and the substrates, those small molecule probes, will be able to penetrate the structures. But it's actually more complicated than that, because if you, for instance, consider that these large spheroid structures actually also have a gradient of oxygen that is able to penetrate, or is actually restricted from penetrating all the way to the middle. And for some of the very large structures, you may actually have dead cells in the middle. But the issue also comes down to, if you have a decreased oxygen content in the center of a large spheroid, that may also restrict the luminescent signal from that region of the spheroid, because both the shrimp enzyme and the beetle or the firefly enzyme also require oxygen to do that catalytic step. So we've done some preliminary experiments using environmental chambers that have, you know, the ability to change the gases in the atmosphere, and you know, we do know that in some cases, very low concentrations of oxygen can be limiting to the luminescent signal. So I hope that partly addresses the question, but it can be very complicated depending on the actual biology of the cells in different locations in these large spheroids.
Yep, thank you, Terry. So, Laura, if you have any additional comments or question, please reach out to us. Thank you. Then another question for you, Terry, is organoids are most of the times hollow structures with a rather limited amount of cells in the lumen. Did you also test your assays with compact 3D structures?
Most of the work that we did internally at Promega used compact structures, usually individual spheroids formed from tumor cells like the HCT116 cell. In those cases, those spheroids are not hollow. We do have people in the field or collaborators that have used our products with the organoids, and I agree that most organoids do have this cyst-like structure where there may be as little as a single layer of cells surrounding a fluid, and in those cases, of course, you know, those are much easier to lyse. But for most of the work that we did, we used the individual scaffold-free spheroid approach simply by adding the cells either to an ultra-low binding plate and allow spheroid formation, or alternatively using the hanging drop method by forming spheroids in a hanging drop for a period of like typically 4 days.
Okay, yeah. And then another question, the metabolite assays are targeting metabolites in the supernatants. Are there also assays for testing metabolites within the cells?
Yeah, so it's actually an experimental approach, you can actually, harvest the cells and wash them in buffer to get rid of the metabolites that would be in the culture medium, and then do a cell lysis step and use that same assay chemistry to measure the metabolites from within the cells. But it becomes complicated, depending on your efficiency of washing away the metabolite and how quickly the metabolite would be able to diffuse from the cells back into the wash solution. But those are controls you would have to set up. But yes, you can use the metabolite assays to measure, medium or extracts or total.
Yeah, I think that answers the question. Thank you, Terry. I have another question for Flavio now. So a person would like to know if it would be possible to perform real-time bioluminescence imaging from the organ chips.
I think with bioluminescent imaging, you need a specialized reader or a specialized microscope. But it's possible to interrogate these chips over time the way we do, for example, for this barrier integrity assays, which at different time points the plate is measured. Or if there's like an incubation chamber, this can be used directly in the acquisition device.
Yeah, and this person is also adding if also luciferase is expressed from the organ models on the chip. So is it?
I think if luciferase is expressed and there's like a constant luminescent signal, then this can be measured on plate as well. So most of this luminescent assay we do sample out and measure in a white plate. However, it's also possible because also plate reader can measure an organoplate, so you can measure it real time. Yes.
Okay, thank you, Flavio. I think we have come to the end. We have 1 hour. So thank you both, Terry and Flavio, for your nice presentations and your answer to the questions. If there are any other questions from the audience, please don't hesitate to reach out to us. So the contact details are in the slides. The recording will be available for you afterwards. The slides from Terry can be shared afterwards. I need to ask Flavio if we might be able to share your slides maybe in the future, but that's maybe something you need to check internally. And I want to thank everyone for its attendance. And just one last note: we have next week another session about nanolog technologies, specifically nanobit and NuMet technology, and then on the 3rd of December about target engagement and protein degradation. So thank you very much, everyone, and wish you all a nice day and stay safe.
Thank you.
Thank you very much.
Thank you. Bye.